Hello, and welcome to the December Research Information Volunteer Call on Disease-Modifying Therapies for T1D, Part 2. Today's speakers are Jay Tinklepaugh, Ph.D., and Josh Vieth, Ph.D.
Hello, everyone, and thank you for joining us for the second edition of the Disease Modifying Therapy RIV call for this year. We're back this month to talk about the other side of the portfolio. Last month, we talked about immunotherapies, and today we'll be talking about all of the work that's going on in beta cell therapies. Now, as a reminder, last month, we talked about it being National Diabetes Awareness Month, and we were highlighting all the ways that we're moving forward for type 1 diabetes in disease-modifying therapies. And we talked a little bit about how we're JDRF-funded active clinical trials are moving the field forward. We talked about investing in improving clinical trials to accelerate the development and testing of therapies moving forward. And we also talked about how we're strategically funding new projects to fill gaps in knowledge and to bring next generation therapies forward. And we're going to continue upon that theme today. Again, we'll touch on the pipeline because really this is the crux of what we do here in the research department. And it's here you can see in the entirety of the pipeline and basically how JDRF works across the development of products from the left side to the right side. Now, our approach, harnessing the strengths of research, advocacy, and community engagement, it really enables us to accelerate work across the pipeline and move as quickly as possible from the research discovery phase to the clinical testing and the getting these therapies into the hands of patients. Again, as a reminder, the three sections of the CURES program are global universal screening, disease-modifying therapies, and cell therapies. And again, you also in research have improving lives and our training initiatives. Today, we'll again be focusing on disease-modifying therapies. And under disease-modifying therapies, our goal is really to work on therapies that include treatments that control the autoimmunity and that preserve or regrow beta cells. We're accelerating the development of these therapies into clinical testing and eventually regulatory approval and access. And so there's primarily two questions we ask in the disease-modifying therapy portfolio. For immunotherapies, we ask, how can we turn off the immune attack on the beta cells? And under beta cell therapies, we ask, how do we preserve and support functional beta cell mass? Now, if we zoom in on the beta cell therapies section of the portfolio, there are really three aims that we focus on. One is to preserve beta cell survival and function. The second is to increase functional beta cell mass. And the third is to create new beta-like cells from other cell types. And we'll touch on each of these areas today. But before I hand it over to Jay to talk through the exciting research going on in beta cell therapies, I'd like to give you a quick clinical trial update. Now, you'll remember last month that I showed this slide. It's the disease-modifying therapy clinical pipeline. It shows all of the clinical trials we're currently supporting in disease-modifying therapies. Today, we're going to focus in on the beta cell therapies, particularly on the beta cell survival clinical trials. And we'll touch on three of these today: denosumab, DFMO, and verapamil. Quickly, though, before we do, I'd like to touch on one other clinical trial that we weren't able to talk about last month, and that is baricitinib. This just reported out this week, and we're excited to share some exciting results from that study. Now, this is from the BANDIT clinical trial, and baricitinib is a JAK inhibitor, and JAK inhibitors are critical to the signaling pathways that are involved in both immune cells and with beta cells. It's currently approved for a range of diseases from rheumatoid arthritis, hair loss or alopecia, and also COVID-19. It's important to note that this is a daily oral pill, and in this JDRF-funded clinical trial, which is, again, published in the New England Journal of Medicine today on the day we're filming this, they showed in 60 newly diagnosed children and young adults that baricitinib is able to preserve beta cell function as measured by C-peptide levels. It also improved blood sugar variability in time and range and decrease the requirement for external insulin. Along with these positive results, it also showed a very favorable safety profile. So we're very excited to see what the next steps are for baricitinib. Now I'm going to turn the talk over to Jay, who's going to talk through some of the exciting projects going on in beta cell therapies.
All right. Hi, everyone, and thanks, Josh, for that introduction. I'm going to be talking about some of the really exciting beta cell projects that we have in the JDRF portfolio, starting with ways to preserve functional beta cell mass. So one of the first lines of intervention that we can employ in T1D is simply to improve the survival and function of beta cells in response to the stress and attacking immune system, effectively identifying ways to preserve working beta cells. So just to remind everyone, we know that during the progression of type 1 diabetes, beta cell stress and dysfunction contributes to additional beta cell death. It's not just the immune system. And JDRF-funded research has really identified pathways in drugs that can keep beta cells alive by increasing their resilience during the immune attack. So in this area, there's been quite a few success stories lately. Historically, by testing medications from other indications, have shown benefits on beta cell health. Notable example of this is verapamil. Verapamil, for those who don't know, is actually a blood pressure drug that was approved by the FDA in the early 80s. And as you can see from the figure on the right, verapamil treatment can actually protect mice from diabetes by decreasing a specific protein in beta cells when they encounter stress. So in this case, it shows that verapamil has a protective effect and maintains insulin production in a mouse model. So this has really driven JDRF to help support and fund 2 clinical trials in new onset T1D, one in adults and one in children, that have helped show that verapamil aims to slow progression by helping maintain C-peptide. And as you can see on the right, those taking verapamil will need less insulin and have better overall glycemic control than those on a placebo treatment. So there's now a clinical trial of verapamil alone and with immunotherapy treatments in the European consortium INNODIA, which is a public-private project co-funded by JDRF. So in addition to repurposing verapamil and moving that through the clinical pipeline, there's also an ongoing trial with doctors Emily Sims and Linda DeMeglio focused on targeting type 1 diabetes through polyamines with DFMO. So in the past, JDRF ran a clinical trial to actually see if a sleeping sickness drug called DFMO worked to improve the health and function of beta cells. And in that study, participants undergoing treatment actually had healthier beta cells with better insulin production compared to those taking the placebo. So this project really aims to test this therapy in a larger number of people for 6 months, relative to 3 in the earlier study, and people with new onset T1D to see if these results hold up and we're able to help sustain healthy beta cell populations. So if successful, the clinical trial will really confirm this novel approach to beta cell preservation and form the basis for additional trials to help delay T1D onset in at-risk individuals. We're also supporting a phase 1 clinical trial with Dr. Rupi Vasavada, focused on examining the impact of an osteoporosis drug called denosumab on beta cell health. So historically, we know that beta cells are injured by a pathway that is also known to cause bone loss. Dr. Vasavada has proposed that we use an osteoporosis drug to help block this pathway and improve the health of human beta cells and protect them against injuries that are relevant to T1D, effectively delaying or reversing the disease. So this will be a phase 1 clinical trial. The drug will be given 4 times a year, and we will observe whether or not it improves beta cell function and glucose balance in people who have early type 1 diabetes. So now I'm going to transition to projects that are focused on increasing functional beta cell mass. So this second approach is really focused on finding ways to increase the number of beta cells that have survived the immune system attack and corresponding beta cell stress. The goal would be to replace the beta cells that were lost by encouraging the survivors to multiply and restore their overall number, as I've shown in the cartoon below. So for many years, beta cells were considered part of a small subset of cells within the body that are considered non-proliferative. By that, I mean, they don't multiply and reproduce themselves. However, thanks to JDRF funded research, there have been several exciting discoveries in this area over the years. One notable example was the JDRF partnership with GNF Novartis that really provided some of the original exciting data suggesting that we can increase the beta cell population in vivo. However, there are some concerns that these strategies are not beta cell specific, and they might actually impact other parts of the body. This is a huge problem because increasing the number of cells in the wrong parts of the body could create safety concerns. And so we at JDRF are supporting projects in this area that include both targeted and non-targeted approaches to the beta cell. So one really exciting project in this area is actually from Dr. Andy Stewart. And he proposes the development of a new DYRK1A inhibitor in preclinical studies to help increase functional beta cell mass. So historically, JDRF research has identified DYRK1A as a promising target to increase human beta cell mass. And in mouse models, the DYRK1A inhibitor harmine actually increased in both human and mouse models, I should say, by 300 to 700% over 3 months. In addition to this, in a small clinical trial in non-diabetic volunteers, harmine was given in doses over 150 times higher than those required to make human beta cells regenerate, and there were no significant safety concerns. So Andy's work here will really support and hopefully drive this approach towards the clinic in a way that we can safely increase the population of functional beta cells. And the last subset of projects that I'm going to be talking about are focused on ways to create new functional beta cells. So this third approach we are talking about involves creating new beta cell mass or new functional beta cell mass from other tissues or other cell types within the body. So we've known for a long time that as type 1 and type 2 diabetes actually progress, other cells in the pancreas try and change into beta-like cells. Unfortunately, not enough of them are able to make this transition to restore insulin supply to the body. However, if we approach this either through gene therapy or non-gene methods, maybe there's a way in which we can deliver signals to other cell types in the body to force them to be more like beta cells, to produce insulin and make up that deficit in type 1. So we're really researching ways to use either the gene therapy or chemical interventions to deliver these signals, and developing ways to both control this approach, but deliver them effectively and in a safe manner to ensure that only the cell types that we want to change are impacted. So a really exciting project in this area comes to us and is being supported by JDRF and the lab of Dr. Stefan Kubicek. And Dr. Kubicek is investigating how pancreatic alpha cells can become beta-like cells through small molecules that induce insulin expression. So previously, with JDRF support, Dr. Kubicek in a genetic screen found 4 proteins that without results in the increase of insulin in alpha cells. So with this knowledge, he is developing small molecules that can effectively downregulate those proteins and hopefully restore insulin production. So just to summarize, currently there are a number of exciting projects focused on preserving, increasing, and creating new beta cell mass in type 1 diabetes. We hope to advance these strategies alongside or in combination with promising immune-focused therapies, some of which you heard about in last month's RIV call, to ultimately prevent, halt, and cure type 1 diabetes. There are still some major barriers to progress in this area, including the cutting-edge nature of the technologies being developed, and honestly, the limited targets and strategies that are specific to the insulin-producing beta cell. However, JDRF is addressing these barriers. And over the past year, we have launched several successful and very exciting requests for applications designed to close these gaps and advance T1D research. And I'll hand this back over to Josh to go through a couple of those.
Thanks, Jay. To wrap things up, I'm going to talk through the current requests for applications that we have in disease-modifying therapies. Now, a request for application is a targeted funding strategy we use. And the primary goal here is to strategically fill gaps in knowledge in the field, in particular interest areas, or to provide funding toward initiatives that can move the entire field forward. We have 3 active RFAs, or requests for applications, currently in disease-modifying therapies, and a 4th that actually just launched today. So the first one I'll mention is developing combination therapies in T1D, and we've addressed some of this in previous RIVs. The goal of this RFA was to identify and validate combinatorial approaches. So how can we use multiple drugs, multiple therapies to provide sustained rebalancing of the immune system and prolonged protection of beta cells? Now, these projects are active. There's 10 of them in total, and they started in 2022, and we're expecting them to report out in 2025. And there's a total funding commitment to this RFA of $5.6 million over those 3 years and 10 projects. The second active RFA is realignment of established targets and mechanisms for delaying or reversing type 1 diabetes. The goal of this RFA was to identify and validate therapies that are being tested or have shown efficacy in other diseases for a potential role in type 1 diabetes, with the idea being that this provides accelerated development and lessening of the time from the discovery research or preclinical research into testing in patients. The project timeline here, those began last year, or actually earlier this year in 2023. And we'll go through 2026. There are 5 total projects selected under that RFA for a total funding commitment of $3.5 million. Finally, related to some of the work that Jay talked about today, there's the development of islet targeting drug delivery strategies for type 1 diabetes. And the goal of this RFA is to take the identified regenerative therapies and drugs that have come up through various JDRF funded research projects and other avenues, and find the targets. So how can we safely get these drugs to the cells they act on, safely to the islets and to the beta cells, to provide a promising therapeutic regenerative approach? And these projects again started this past year in 2023, and will go through 2026. There were 5 projects selected under that RFA for a total funding commitment of $4.4 million. And as I mentioned before, just launched today is an RFA titled Development of Interventions to Modulate Immune Cell Trafficking in Type 1 Diabetes. One of the major components of the autoimmunity that occurs in type 1 diabetes is the trafficking of these reactive immune cells, or T cells, to the pancreas, where they can attack the islets and beta cells. We're looking for projects that can identify ways to modulate this trafficking, to block those cells from entering the pancreas as another way to stop this autoimmune attack. This could potentially be a standalone strategy to rebalance the immune system, or to change the trafficking of the cells that other drugs such as Tzield or teplizumab or baricitinib can act on to increase their efficacy. So we're very excited to see the projects that come in and are submitted under that RFA, and we'll give you a full report on what's selected and where we're moving in that direction when it's available. So looking forward, as we've now had 2 RIVs in disease-modifying therapies, where are we going and what are we looking forward to over the coming year or so? One, we're looking to identify discovery research that focuses on new and expanding areas of immune intervention, included with the new RFA I just spoke about a second ago. We're looking to continue the development of combination therapies that address both the immune dysfunction and the beta cell function. So taking the 2 parts of the disease-modifying therapy portfolio and putting them together to a sustained therapies that can benefit patients at all ages and all stages of disease. We're looking to expand the number of targeted drug delivery approaches we have, both for targeted delivery to the immune cells that are causing disease, but also directly to the beta cells in the case of regenerative medicine. And finally, we're looking to drive discovery research into the clinical setting with the goal of, as always, developing a cure for T1D. How can we take the discovery research efforts and the preclinical efforts and move that through the clinical pipeline to take these advances and get them into the hands of patients? With that, I would like to thank you all for joining us for the second Disease Modifying Therapy RIV call. Jay and I both look forward to the live call next week and answering any questions you may have. Thanks.